Hello, my name is Jessica Winsell, and today I will be discussing the KRAS protein and the G12C mutation to my audience, who will be my friend in high school who doesn't have much experience with biology or chemistry. So, what is a protein? Proteins are molecules that do a lot of work inside cells. They are made of amino acids linked in a chain that fold into a specific three-dimensional shape, which determines their function. The protein that I'm going to talk about, called KRAS, would be classified as a type of relay molecule that helps control when cells grow and divide. It receives signals from the receptors on the surface of a cell and passes those signals along to other molecules inside the cell. Normally, this protein acts as a switch that turns on and off depending on when the cell needs to grow, but mutations can cause the cell to be stuck on the on position, which keeps cells dividing even when they shouldn't, which can lead to cancer. Proteins have different levels of structure. This is a primary structure, which is the sequence of amino acids in a protein. Here I modeled a short sequence of 12 amino acids at the beginning of my protein, but it's important to note that the whole protein would be much longer, with 189 amino acids. At the position of the 12th amino acid in the chain, the amino acid glycine would normally be located. This is the blue G right here. And when the G12C mutation happens in the protein, this glycine is replaced with cysteine, which is this green C right here. This change occurs very early in the protein sequence and influences how the protein folds, ultimately affecting its three-dimensional or tertiary structure and its function. This model represents the tertiary structure of the KRAS protein, which is a three-dimensional shape that the protein forms after it folds. The KRAS protein is attached to the inner surface of the cell membrane, and this protein is found in many different cell types in the human body. In my model, the yellow color pipe cleaners represent the 6 beta-pleated sheets, which are where the protein chains fold back and forth like a zigzag. And the pink pipe cleaners represent the 6 alpha helixes, which are where the protein folds into spirals. These helixes and sheets are examples of secondary structure, which are smaller folding patterns within the protein. When all these secondary structures fold together, they create the full three-dimensional globular protein. Some proteins also have a quaternary structure, which is when multiple protein chains join together to form one functional protein. However, KRAS is just made of one polypeptide chain. Here we can see the position of the amino acid glycine in blue in the normal functioning KRAS protein. The activation of KRAS works like a switch using two molecules called GDP and GTP. In my model, the green ball represents GTP, and when it is bound to the protein, it is in its active state, sending signals telling the cell to grow and divide. When the signal needs to be turned off, KRAS turns GTP into GDP, represented by this red ball, which turns the protein off and causes it to enter the inactive state. Normally, KRAS switches between these two states, but when the G12C mutation happens, glycine is replaced with cysteine, as seen right here. Glycine is a small and hydrophobic amino acid, which means it avoids water, but in contrast, cysteine is a large hydrophilic amino acid, which is more reactive and can form additional chemical interactions. Because cysteine behaves differently, this change alters the shape and behavior of the region of the protein that helps control the switch between the active and inactive states. As a result, KRAS has a harder time turning off, which means it can stay in its active GTP-bound state longer than it should. When KRAS stays active for too long, it keeps sending signals to tell the cell to grow and divide. This is why mutations in the KRAS gene are strongly associated with cancer. So even though the mutation changes just one amino acid, it can have a huge effect on how the protein behaves and how the cell functions. Although this model does help to represent the overall structure, it has several limitations. First, the KRAS protein is much more complex and contains many individual amino acids that are arranged very precisely, which my model with pipe cleaners cannot fully represent. Second, the real binding site where GDP and GTP attach has a very specific shape that fits these molecules precisely. In my model, the balls representing GDP and GTP only show the concept of binding, not the exact structure of the binding site. Third, the mutation from glycine to cysteine changes the chemical property of the amino acid, but my model only shows the location change, not the specific interactions that actually cause functional and structural change in the protein. Despite these limitations, my model helps to visualize how the KRAS protein folds and how small mutations can affect its function.